I think we'll go ahead and get started. I'm considering we're a few minutes after now. We might still have some folks roll in, but we have a couple of housekeeping things to get through first. So welcome to everybody who's joining our webinar today. I see in the chat we have people joining us from all over the world. I'm always interested to see, what kind of time zones people are in. So I guess good morning or good afternoon or good evening, depending what part of the world you're joining today. And I'm glad to have you here. So I'll go ahead and open by saying my name is Veronica Obregon Perko, and I'm an application scientist on the FlowJo team with BD. And in today's FlowJo webinar, we're going to be talking about supervised auto gating tools in FlowJo software. So what we'll learn through the course of today is alternatives for manual gating, but also alternatives for completely unsupervised clustering, and talking about tools that sit somewhere in the middle of that spectrum. So I'll open with some slides just to kind of set the stage for the topics we'll be talking about. And then I'll spend the rest of the time in the software just quickly demoing what these tools look like, where you can access them, and how you can install them. Okay, so shouldn't go more than an hour today, might be a relatively short webinar. But that means there'll be plenty of time for questions. If you do have a question, you do not have the ability to unmute. And if you raise your hand, I also can't unmute you. But what you can do is go ahead and put your question in the chat or in the Q&A box. And I'll do my best to keep an eye on those throughout the presentation. You don't have to wait till the end. You can raise the questions as they come up, and I'll do my best to address them when they're relevant. And this webinar is being recorded, so if at any moment you have to step away from your computer or you want to share some of the content you learned with your lab mates, I'll show you where you can access that recording, but it essentially gets posted to our website, usually within a few days after the event. So you'll be able to rewatch this session, and I'll point out some other learning resources on our website as well. Okay, so with that all being said, let's go ahead and get started. So I think in the era of, big data, our flow cytometry experiments are getting big, right? And that means that so is our gating, right? And the amount of time we have to spend gating all of our data. So this is just to kind of show you, what I have in mind when I think about the evolution of flow cytometry, really since the advent of that technique into the field. So, if we look at these two NK panels, it's clear to see that panels are getting bigger and bigger, right? And more panels bigger panels means more colors or more populations to identify. So if you take, for instance, these two OMIPs, which are published peer-reviewed panels, there's a 14-color NK cell panel and a 27-color NK cell panel. And you can see with the 14-color panel, maybe within 20 gates or so, I can identify most of the phenotypes that are going to exist in that panel. Whereas if I go to the 27-color panel, right, that is monumentally bigger, and that's going to be a lot of complex immunophenotypes that exist in that sample, right, when I stain it with this panel. And here it's kind of oversimplified because you're seeing single positive gates for all of these different functional markers, but we're not even talking about double positive, triple positive. So whether you choose to gate those manually or use clustering tools that we'll talk about in a second, right, that's going to be a lot of populations to inspect and look at and manually gate yourself if you do go through that approach. And even if your panels aren't that big, let's say they're a more manageable size, like less than 10 colors, our sample sizes are also getting bigger. We have cytometers now that are set up to do more high throughput screenings. So rather than, running a handful of fax tubes, you can run whole plates. And so now you're getting, hundreds of samples, getting the data from those, versus getting sample data from, individual tubes. So really we're dealing with, larger panels. More panels means more populations, right? And we have more samples. All of that just adds up to more time finding gated populations, right? Whether that means that it's a large panel or just because you have a lot of samples. As flow cytometrists, we spend a good part of our analysis just drawing gates and moving those gates around. Right, so what are the approaches we can take when we need to identify populations in our samples, right? No matter how big the panel size. So there's, of course, the traditional approach, which is just to define populations manually, right? So a supervised gating approach. So in this case, your populations are user defined. and you're usually following some kind of biology that's established or that's established in the literature or in your laboratory. And you go into a flow cytometry gating strategy, something like this, right? You have an idea that T cells are always CD3 positive, so you start there and truncate off that, knowing that CD8 T cells are, mutually exclusive for CD8, and they don't have CD4, and vice versa for CD4 T cells. And you know that different combinations of memory markers lead to different combinations or different subsets of memory T cells. So when you follow a gating strategy like this, then it's user defined, it becomes easy to align phenotypes with established biology, right? When you find a population and it's straightforward to say what it is because you used, well-defined markers to get to that population. But when we talk about a traditional manual approach, right, there are some considerations. One being that you have to have required knowledge of the expression patterns for the populations you're looking for, right, which sounds kind of obvious, but that can be a little more nebulous as we dive into these high parameter panels where you're perhaps trying to explore novel phenotypes. And so if you're looking for something novel, that means you don't know what it is. You don't know what it's expressing, to what level, and in what combination relative to the other markers in the panel and the other populations that exist. So it doesn't lend itself really to a discovery-based approach. So you have to come in with some kind of a plan on what you're gating for and knowing, what those cell types are expressing. So even if you were still okay with that, let's say you have a relatively straightforward population identification strategy, like what we're seeing here in this example, you'll ultimately still need to draw those gates and apply them to all of your samples. And I think we're all familiar with the fact that the gates will have to be nudged, right? Even in FlowJo, it's very easy to copy gates from one sample to another or to a group of samples from another, but that just places the gates onto the sample. That doesn't mean they're going to be in the appropriate place, right? And then we can kind of see that in action down here, right? Very simple example where I have sample number 1 and I've drawn a gate to capture this population in the bottom right quadrant. But when I drag that gate onto my next sample, right, I've drawn the gate, but it's not exactly in the right place, right? The gate probably needs to be a little bit bigger, maybe needs to shift over to the left a bit. So this population has not only changed kind of in shape, but it's also shifted along the axis for that parameter. So some of you might be thinking, well, I use magnetic gates in FlowJo anyway, so that kind of takes care of a lot of those problems of population shifting. So for those of you that are not familiar with magnetic gates in FlowJo, it basically gives you an opportunity to draw a gate, make it magnetic, with the check of a box, and then that gate will attempt to move with the population as it varies in placement between samples. And it's true, it definitely will solve a lot of these problems, but it's not perfect. It's going to track shifts in populations, but not necessarily shape changes, right? So even if I had made this magnetic gate here on sample number 1, the gate may have shifted to the left a bit to kind of capture that change, but it may not change the shape of the gate. Or it won't change the shape of the gate. So it's not going to capture always the shape of that population. So if the population has gotten bigger and now it's outside of the gate, or the population is much smaller and now that gate is overdrawn, it's too big, it's not necessarily going to change the shape. It's just going to try to catch the center of that population. So it will do a lot, but it won't always capture everything. Now, an alternative to manual gating, and it would be a strong alternative, would be unsupervised clustering. So for those of you that have watched any of our high parameter workflow webinars, we talk about these tools very heavily. So I'd encourage you to watch one of those webinars if you're very interested in these tools. But just to summarize it on a slide, defining populations automatically, can be summarized as basically unsupervised clustering. So ultimately with these tools, your populations are identified through computational algorithms where clusters are formed, and these clusters are going to represent phenotypically similar cells, right? And we can see that in action in this example here. This is a published clustering tool called Phenograph, and here's the publication down here. And essentially what the authors have done in this example here is taken some data, run it through this clustering tool called Phenograph. And Phenograph has identified 15 different clusters, right, or 15 different populations. Now, it doesn't tell you what they are, right? It just numbers them 1 through 15. And if you run a tool like this in FlowJo, that's essentially what you'll get, just 15 different populations that are labeled numerically. But what the authors propose in this paper is that, if we annotate this same data, and we can find phenotypes that basically can be found manually that match to these clusters, right? So these clusters are found automatically, right, without supervision. And they can be traced back to, meaningful immunological phenotypes that we can find in the data manually, right? And here's kind of the cluster and like a phenotype it would match to if we gated the data manually, right? So they are finding meaningful populations most of the time. And so it can be an unbiased and fast way to create an opportunity for discovery, right? Give you an opportunity to not only quickly find populations you already knew about, but also maybe find populations you hadn't thought of, right? Combinations of phenotypes that you are unaware of, and you can reveal that in your data by just kind of letting the data drive the identification of those populations rather than you taking a manual gating approach. Now, considerations for unsupervised clustering, and this is for any unsupervised clustering tool, not just Phenograph, right? Sometimes the clusters can be hard to interpret and annotate, right? If you have a really large panel of say 30 to 40 colors, right? Those 30 to 40 parameters have all been considered in the formation of that cluster. And so you may get combinations of markers that don't make sense initially, right? These clusters have just been identified as informatically different from the other clusters, but that doesn't mean that they're going to be straightforward for interpreting the biology, right? You might not understand why a cluster is expressing certain markers, and they also need to be inspected to make sure that clusters have separated appropriately, and you don't have clusters that need to be merged. So at the end of the day, these algorithms are not biologists, so you have to help that biology along and understand if a cluster is biologically relevant, or if it's some kind of erroneously separated cluster that doesn't need to be pursued any further. And again, the clusters ultimately have to be annotated, right? The tool is going to return the populations numbered. For this example, they're numbered 1 through 15, and it's up to the user to go into the clusters and start to give them some kind of identity, right? And explore what they're expressing. And we have tools to help with that, but it is still something that needs to be done. And at the end of the day, you don't get any resultant gates for adjustment, right? So if you're unhappy with the clusters that come out the other end of the tool, right, unhappy with the populations it's found, you can tune a couple things in the algorithm to increase or decrease the number of clusters you get back, but that's pretty much all you can adjust, right? And that's why we call these tools unsupervised, right? You can't sit there and tell the algorithm what you think a CD3 T cell is, what you think a CD8 T cell is, what the boundary of MFI cutoff will be for the separation of any cluster for a given parameter, right? So that's why it's called unsupervised, because you don't get those opportunities to input that information, right? You're just kind of letting the data speak for itself. And so again, while it's unbiased and fast, it doesn't offer a lot of flexibility for what the outputs are going to be. Okay, so in that way, it can be unpredictable in the sense that, through multiple runs, you might get multiple different results, and you ultimately have to dig into those clusters and identify what they are. So to summarize the two previous slides, right, what are my options then for gating, right? And particularly in FlowJo, right, we talked about these two slides, and they can kind of be summarized onto this spectrum where I've put, supervised tools on the far left and unsupervised on the far right. Right, so we have manual gating, which is, again, ultimately user-defined, right? That's going to require user knowledge of biological phenotypes that they're looking for in their data. And of course, we have a lot of different gating tools in FlowJo to help you capture, right, the shape of your data, everything from a rectangle to a freeform polygon shape. And so that's going to be, of course, 100% supervised, right? That's you dictating where the boundaries of a population begin and end. And on the other far side of the spectrum, we have unsupervised tools, right, where you're basically letting the data speak for itself and kind of trusting this algorithm to find these populations for you, right? And these are typically in the category of clustering tools, where you're computationally defining populations, right, and allowing those populations to be segregated in the high-dimensional space based on their expression patterns for their markers. And you don't get an opportunity to help that tool along and tell it, what's positive, what's negative, what's a T cell, what's a B cell. And inside FlowJo, we have 3 of these tools available for you to use as plugins. There's Phenograph, FlowSum, and XShift. And again, we have webinars on these topics if you'd like to learn about these tools a little bit more. So could there be something in the middle, right? Maybe I'm okay in trusting an algorithmic approach to help refine my gating strategy, but I don't want to just get a bunch of random clusters that I have to annotate. I do still want to offer guidance based on a manually defined gating strategy. I want to be able to say what I think a T cell is or where I think a gate should go. And so can there be kind of a sweet spot, right? A marriage between manual supervision and kind of letting these computational tools, take the wheel for at least some part of the analysis. Right. And I'm sure you could have guessed the answer is yes. And that's why we're all here today. And so this will be the focus of the rest of the webinar, is something to be called, supervised autogating, right? Or semi-supervised clustering. Basically, the computational gating of user-defined populations. Okay? So these are the 2 tools we'll be talking about, both available as plugins to be used in FlowJo software. We'll talk about EasyDaffy and BD Elasticate. And what we'll learn about these tools is that they kind of sit somewhere in the middle of manual gating and clustering, right? An opportunity to instruct the algorithm on what to find and give you an opportunity to simplify your analysis so you don't spend so much time doing manual gates or spend so much time annotating clusters that, although were found very quickly, you still have to do some homework on. Right, so let's talk about Daffy first. So here's the publication if you would like to learn more about the tool and how the algorithm was designed. But it stands for Directed Automated Filtering and Identification of Cell Populations. So ultimately, it's an algorithm that starts with a manual gate and adjusts its boundaries via clustering. So you can think of it as building your own personal clustering tool based on manual gates that you've defined, right? So let's talk about the schematic down here. So basically the way it starts is here's your input data in black. Your data will get clustered in the background, right? And it's going to cluster using a self-organizing map or k-means. And it does this clustering in the background. So here's each of my clusters, right, identified with each of these different colors. And then here's my manual gate that I've drawn, and it's going to include 2 clusters, right, that were identified with clustering, but I'm setting this manual gate boundary, right? So I want to capture all the cells inside this box. And what the algorithm sees is that inside that box, I have a lot of the blue cluster, a lot of the pink cluster, and a couple of the purple, but most of those purple events are kind of outside of that boundary. Okay, so we're going to say that basically it seems like Veronica's trying to capture in this gate everything that we would have identified as a blue cluster or a pink cluster. Okay, and so now we're going to unify those clusters and call them, my special cluster, which I defined with this manual gating boundary. And the algorithm basically continues in a recursive fashion until it does this for all of your manual gates. Oh, and it looks like we have something in the chat here. Oh, yes, everybody's still saying where they're from. Super fun to see where everybody's joining us from. And so that's basically it in a nutshell, right? Clustering in the background, you manually define which clusters or I should say, which populations you want to identify in your data. And then those clusters get refined, right, to capture that boundary that you have set with a manual gate. So some notes about the operation of the tool in FlowJo. So what you get at the end of running the tool, and we'll see this in the demo, is essentially you get clusters, okay, but you're not getting adjustable gates. It's going to behave much like a unsupervised clustering tool would, like Phenograph or FlowSum, right? You'll run the tool and it'll return subpopulations for you, but you don't necessarily get gates to be able to adjust anything. So you kind of just take it for what it is, but you are getting that opportunity to supervise the clustering right at the very start by using these manual gates. You can also get an overlay of plots to review the results, because you're not completely blind as to how it performed relative to your manual gating. As we'll see in the demo, you'll get an overlay of the EasyDaffy defined population and how it compares to the population you manually gated had you pursued that approach. And so the plugin's going to need parameters with a stain name. Okay, so it doesn't pass in its current form using scatter parameters. Okay, so if you want to use the tool, you might want to pick a population that's past your cleanup gates, because we can't feed in scatter parameters into the tool, at least for the plugin implementation. All right, so moving on. Trying to advance my slide here. There we go. So another approach would be to use BD Elasticate. This is a relatively new tool to the FlowJo exchange, and it kind of comes at supervised autogating from a different approach, and is really just trying very hard to basically just automate the gate nudging you would do manually, right? So rather than returning clusters, right, that are being used to define populations that you have helped along with manual gating, this is ultimately just trying to copy your manual gating onto new samples, right, and save that headache of moving gates around. Let's see. Oh, there is a question in the chat. How many parameters can you use in Daffy? That's a great question. I'm not aware of a upper limit on how many parameters you can use with EasyDaffy. I'd have to look into it. I've seen it used with pretty high parameter panels, so I imagine it doesn't have a limit, or it's a very high limit. So as long as you're using parameters that have stain names, so like fluorescent parameters mostly, or if you're doing mass cytometry, as long as those parameters are labeled, they should be able to be used in the tool. You just can't use scatter parameters. And I'll show in the demo that even though your panel might have a lot of parameters in it, the tool may ultimately only use a handful of them to render the clustering. It's going to find the clusters that define the populations the best and use those parameters over others. Okay, let's see. There's a follow-up question here. Let's see, there was another question. Oh, so no spectral parameters, only unmixed stainings. Moritz, I would say that you'd probably want to use the I think you can use spectral parameters. I would just make sure that they've been compensated, and I think maybe that's what you're getting to with the unmixed stainings. So I would probably use compensated parameters. It doesn't matter if they've come from a spectral instrument or a traditional instrument, if that is your question. There was another question I think came out. Is there a publication for the technical details of Elasticate? We don't at the moment. We are working on it. It's actually a relatively new tool. So this is the most details I have on it at the moment. And we're probably going to work on, releasing more technical notes in the future. But if you have any specific questions, feel free to email me and I can try to answer them for you. But the short answer is no, we don't have a publication on the tool at the moment. All right, I think hopefully I didn't miss any other questions in the chat. Let me check the Q&A box here. Let's see, there's a question here in the Q&A box that says, for Elasticate, do you need a training set for each condition / treatment? Gina, that's a good question. I will get to that. But the short answer is no, you don't. There will be a setting to try to circumvent that. But you could, in theory, do separate trainings if you wanted to. But we do have a toggle in the tool to address maybe differences in population distribution because of like a biological condition like you described, like unstimulated and stimulated. All right, so I think I am caught up on questions. Let me make sure. Okay. All right, so let's get back to Elastigate. It looks like we've answered a couple questions on it already. But again, essentially taking a different approach, right? So rather than customizing a clustering tool to refine your gating like the way EasyDaffy does, right? Elastigate is basically just really trying to, copy the gates you've already drawn onto your new samples and just help you nudge them a little bit in either direction. So here's a schematic on how that tool is working. So it's basically an algorithm that represents your plots as a picture and then compares the pictures of new plots to those of the training plots that had the predefined gating. Okay, so essentially we're seeing that here. So here's the training data, for instance, right? It's simple, whole blood staining. And so I have my training data and the target data, or the new data, that I'm going to ultimately want to apply the predefined gates to. So it looks at the training image and basically tries to adjust the image to match the target image, right? So how would I have to shift that original picture to match the new picture, right? The new sample. And then once it has that transformation, it uses it and applies it to the predefined gate, right? So if I know that, oh, this new plot is, this target population is now a little bit more to the right and it's a little taller, it's ultimately going to apply that same change to the predefined gate so that the gate will move, right, and now be able to snap to the same target population, but now in the new file, right? So basically treating your plots as pictures. How do I change that picture to match the new picture and apply that change in the form of a gate to the new data? Okay, so in terms of operation notes, right, what happens when you run the tool? Okay, so ultimately the calculated gates are going to get drawn onto the samples, but you can still manually adjust them. And I think this is what sets the tool apart from other auto gating tools that might be out there, is that you basically get those same gates copied onto your new samples. And if you're unhappy with where they've been placed, you can always review them and still move them around in terms of shape and also shift placement on the plot. Okay. And in this tool, you can use scatter parameters, as with the EasyDaffy plugin at the moment doesn't handle those. All right, so setting up the tools in FlowJo. Okay, so for either tool, whether it's EasyDaffy or the BD Elasticate plugin, you can retrieve each of those plugins from the FlowJo Exchange, right, at no additional cost. You just need your FlowJo license to be able to use them within FlowJo. So you download those tools. When you download them, they'll come in a zipped folder, and inside the zip folder, you will have a jar file, which is really what FlowJo needs. So you'll take that jar file and just drag it right inside a plugins folder. Now your plugins folder, you on a PC, you get one made for you in your program files when you install FlowJo. So you could just use that, or you can create a folder somewhere locally on your computer, right? Name it plugins, and then just put your jar files inside there. On a Mac, you can create a folder in your applications and call it plugins. And again, drag the jar file right inside. And then you just open up your preferences window in FlowJo, go to the diagnostic section, and you can just tell FlowJo to scan for a plugins folder and tell it where that folder is. And I'll quickly go through this in the demo. Now, I will say, if you want to use EasyDaffy, it's got a couple extra dependencies because it is an R-based algorithm. Okay, so if you want to use EasyDaffy, you'll also need to install R and set that R path in FlowJo, which for a PC typically looks something like this. And then you'll also need to install one other dependency, right? So you'll need R for sure. And then if you have a PC, you'll also need R tools. Okay, and then you also have to put R tools on the path. I'll show you how to do that. It's actually quite simple. And if you have a Mac, you'll need to install Xcode or Xquartz. Okay. Now, just a reminder that BD Elasticate doesn't need any of the tools listed here at the bottom, but if you are interested in EasyDaffy, you'll need to install R and R tools or Xcode. All right, so let's go ahead and move into the demo. Before I go into the software, let me go ahead and just take a field trip to our website so I can show you where to get these tools. So if you go to flowjo.com and navigate to downloads, FlowJo Exchange plugins, okay, you'll be able to search for any of the tools you're looking for. Okay, so in this case, if I'm looking for EasyDaffy, okay, I can search for it and it'll take me to the landing page. I'll also show you that if you don't hit the search, right, you can also just search through these cards, right? And so for instance, if I'm looking for Elasticate, okay, so here's the information card. Okay, it'll give you a summary of what the tool does, and then you can download the most recent version. For any of our plugins, if there's any kind of compatibility issues with our versions, we typically list that right here. So when you download that tool, okay, what's going to happen is it's going to download a zip folder. Okay, in that zip folder, you'll open it. And actually, let me just open another one instead of waiting for that one to download. When you open the zip folder, right, you'll see this .jir, this .jar file, and you'll drag that into the plugins folder that you've either created locally on your device, or you can use the plugins folder that comes with the program files when you do a PC install. right? So that's where you can download the tools and move the jar file. For those of you interested in more education and the recordings, if you go to the Learn tab, Webinars, okay, here you can see this is where you registered for the webinar you're currently watching. If you go to recorded webinars, right, the recording will be posted here, as well as all of our other webinars. Okay, so if you are interested in unsupervised clustering tools, if you watch any of the high parameter workflows, they'll likely go over it. If you want to do a little bit more focused learning on just EasyDaffy, right, we also have a webinar on that topic as well. And if you're ever lost on how to install plugins, if you go to FlowJo Software Help, and just type in something, or actually, I think we have a hotkey for it here, plugins. Okay, we'll get a video showing you, how to install plugins, how to set the different path types, how to execute them. Okay. All right. Now, I also mentioned that you are going to have to install some dependencies. Okay, so you'll need to install R if you want to use EasyDaffy. Okay, you can get that from the CRAN repository here, and download R for either your Mac OS or Windows. If you're on a PC, you'll also need to install R tools. Okay, and then this can also be retrieved from the same resource. I just typed R tools into the search engine, and it was one of the first pages that popped up. Okay, so you'll install R tools and then scroll to the bottom, and it gives you instructions on how to put R tools on the path. Okay, so all you have to do is open R or R Studio, and you'll just copy and paste this code, and that's pretty much it. And it usually sets itself up after that. It gives you some steps to test that to make sure you did it correctly, but I've done it lots of times, and it typically works out fine. Now that's for PC, for R tools. Okay, then for Mac, you'll need R, but you'll also need Xcode and Xquartz, which again, I typed it into the search engine, brought me right to this page, and you can install those extra coding bundles. Right, now let's open FlowJo Software. Now, I'll also, before I go into the demo, show you the diagnostics window. So if you go to the preferences, which is this heart-shaped symbol here, okay, you can open up that window. And if you go to the diagnostics section, okay, this is what you need. So if you're going to use EasyDaffy, you'll need to install R, set the R path here. So basically navigate to where that R was installed. You can also copy and paste paths from our website if you don't want to type it out. And then whether you're using EasyDaffy or BD Elasticate, okay, those are both plugins, so you still have to scan for plugins. So hit this checkbox and then navigate to the folder right where you stored those plugin jar files. Okay. And then usually at that point, I restart FlowJo, maybe restart your computer and give everything a chance to kind of reset and install. And typically the plugins will take it from there. All right, so now having gone through, what these tools can do for us, how can we install them? Let's do a quick demo. So let me turn off my camera for just a second so I can have some more room on my screen. Okay. All right, so let's start with EasyDaffy. Okay, so I've set up a workspace here to get us going. But essentially the situation is I have these 3 samples, okay, LD1, 2, and 12. And I have a simple gating hierarchy here that I've drawn on one sample, and I have kind of really focused attention on putting the gates where I want them to be, right? So if I go ahead and launch, just to kind of show you what the strategy looked like. Oops, wrong one. There we go. Right, so just, I did some cleanup gating on the file, and ultimately, was looking for, live CD3 T cells. I looked at 2 different T cell types, and then ultimately went for some kind of CTL like CD8 T cell based on perforin and interferon gamma co-expression. Okay, so I drew the gates on this sample and really paid attention to their placement, but I don't want to necessarily spend too much time doing that on the subsequent samples. So I've copied the gates onto them, but I don't want to just spend all this time nudging them around. Okay, so in that case, what I could do is just go ahead and create a separate group and drag in that sample where I did this kind of exemplar gating, right? So the reason I'm separating them out is, we'll see this in a second, but you can pretty much only run the EasyDaffy calculation once. So if I separate it out now, I can group apply it to another group where that sample is missing and not run into any problems. Okay, so basically make an exemplar group and have that sample where you predefine the gating strategy, right? And then after we run the tool, I can drag it onto the group with the remaining samples, because if I had the exemplar sample here, it would try to recalculate again and then run into some problems. Okay, so usually better to separate it out. If you don't want to separate it out, you'll have to copy and paste the node individually to your multiple samples. Okay, so up to you. All right, so here's the file that I want to use. Okay, so very important with EasyDaffy, before you launch the tool, go ahead and save your workspace, and that way it saves those gating locations. Anytime you make a change to the workspace or nudge a gate around, right, we need to save that change to the workspace so that EasyDaffy has the most up-to-date calculations to do the clustering. Okay, so if you come in and decide you want to move a gate real quick, just make sure you save it before you launch the tool. Otherwise, it's going to have the last reverted save. Okay. All right, so in this case, I want to go ahead and refine the gates for everything after my live CD3, right? So my 3 different T-cell gates here. So I'm going to pick that parent population, go to Workspace, Populations, Plugins. Okay, this is where all of my plugin tools are. And then we'll go to EasyDaffy. Okay, so here's the dialog window. First choice by default is going to do the clustering with a self-organizing map, which typically does better for more complex data. K-means is also a good clustering tool. It might be good for very straightforward data, right, where markers that are kind of on, off, and you don't have a lot of complex phenotypes, it might be fine. So if you do decide you want to use K-means, just uncheck that box. Otherwise, it's going to use SOM by default. Create a summary layout, right? Take it or leave it. I like having it because I want to be able to see, what that algorithm was doing to my samples. Okay, and if you want an overlay of the EasyDaffy population, you can check that box as well. Last option here, if you hover over, it kind of tells you what it's doing. But essentially the tool is going to only use a handful of parameters to ultimately define the clusters for these manual gates that you've drawn. But if you want it to consider, all of the different dimensions, then you can hit the checkbox. But I will say that it may not always be reliable and it might take a lot longer to run. So typically the 3 that it picks with the highest discriminatory power does a pretty good job. So I'm not going to check this. Okay. And last option, that's just if you're using single cell RNA-seq data, which we're not. This is just flow cytometry data. Okay. So I'll go ahead and say okay, right? And you'll see that it's going to calculate on that live CD3 population, not going to take too long. So right now what it's doing is considering, right, the manual gates that I've drawn, everything that's under that parent population, and basically trying to use, that supervised clustering to find me those same cell types, right, without manual gating. And that way I can apply that kind of custom clustering tool to my other samples, and it's going to find those same target populations, but this time using clustering instead of the manual gates. Okay, so I'm going to let it calculate here for a second. Shouldn't be too much longer. The other thing it'll do when it's done is it'll create a summary in the layout editor, and that way it'll show an overlay of that population that it found on top of the manually defined population. All right. I guess I'll pause here and see if there are any questions so far on the EasyDaffy tool. Nope, I see something in the chat. Let's see. How long does it take for larger experiments, more cells, more files, for example? Yeah, Lucas, that's a good question. I will say at the moment, it's taking longer than it normally takes for me. I do find when I do screen shares, for some reason, my FlowJo doesn't always keep up. I don't know if I'm just using too much power on my computer. So I will say it doesn't normally take this long. I demoed it just before the call and it was going faster. But yeah, I think it's marginal, right? So, I mean, with any of our tools, the more parameters you have, the more cells you have, the more files you have, then yeah, I mean, some plugins will take longer. But I think you'll only really notice a difference if you have really massively different files, right? So if you're running the tool on a panel that has 3 colors versus a panel that has 40 colors, I think there you'd notice a difference, but maybe a difference from like 10 to 20, you probably wouldn't notice that. Okay, this one is taking some time to run. So what I'm going to do is open a workspace where I had this. Well, of course, as soon as I did that, then it finished. But I do still have a workspace where it finished already. But in any case, right, of course, now it's completing everything as I threatened to open a completed counterpart. But we can see the tool's finished running, right? And I promise it doesn't normally take that long. I will say also, just as a heads up, for anyone who uses R-based plugins, the first time you run it ever on your computer, it probably will take a few extra minutes than normal because it's trying to install all of the packages it needs. So you may have to wait a little longer the first time, but typically every time after that is faster. All right, so here's what it's doing now. Okay, so there's the EasyDaffy node, right, that ran off the live CD3. Here's my manual gates, right, CD4, CD8, CTL, and then here's the EasyDaffy defined versions of those populations, right? So if we look here in the summary layout, okay, so here's the manual gate for CD8 T cells. And then there's the EasyDaffy defined CD8 T cell in red, right? So EasyDaffy basically thought like, hey, I would have included all of the cells out here. So that is now your EasyDaffy CD8 T cell population, whereas I would have only captured what was inside the manually drawn gate. And so you're getting that same representation, but for your other populations, right? So here's my CD4 T cells, here's the CTL gate, right? So same thing here. It's like I drew this gate, but using, considering other markers that are even outside this biaxial plot. So not only considering perforin and interferon gamma, but also considering CD8 and CD38 and even viability dye, right? It's saying that it thinks that the CTLs, go a little bit beyond that gate, right? So now it's defining my CTL population, right, as everything in red, and it's going to use that same criteria when looking for that population in future samples. Okay. So beneath each of the gates, you can see the markers that it considered when making those boundary decisions. Okay. And so it typically considers the 2 that are already in the gate, of course, and then 3 additional, right? So in this case, I have CD4, CD8, but it also considered perforin, CD38, and interferon gamma, just to define what a CD4 T cell is. Okay. All right, now at this point, right, so I've done kind of the training, right? I've identified those clusters off the exemplar sample. So it would be a matter now of just taking that node and dragging it right onto either an individual sample, or I can just drag it onto that same population in a group of samples that I want them all to inherit this kind of clustering, right? So they need to have the same gating strategy, so you can apply it to the same node. Now, in this case, I will go ahead and open a workspace where that's completed, because if I drag it onto those 2 samples, each one's probably going to take 1 to 2 minutes to finish. So I'd rather just kind of show you what it looks like, right, with the time that we have. So let me go ahead and do that. Right, but it'll run in the same way, right? So here is the completed version, right? So I basically took that EasyDaffy node and popped it right onto that same parent node, right? Live CD3. And it basically did the same thing, right? There's the EasyDaffy defined versions of those gates. Okay? And if I go into the layout editor, right, it's also going to give me a summary for each of the samples if I check that box in the dialog window, right? So again, here's it showing, what it defined as a CD8 T cell versus what the manual definition was, again, for CD4, again, for CTL. Okay, now a reminder, I didn't really nudge these gates around because I didn't want to spend time doing that. I wanted to use EasyDaffy instead. All right, so that is EasyDaffy. So I think now what I'll go ahead and do, let me close these workspaces and let's try Elasticate now. All right, so once again, I've got a demo workspace for us here, but it'll probably look familiar because it's the same data, right? In this case, I'm just using a different tool. Okay, so now the use case here is that I've got this gating hierarchy, and I basically want to copy that gating hierarchy onto the other 2 samples, but I don't want to nudge gates around. Okay, so that would be the use case for Elasticate. Now with Elasticate, you really only have to draw the gating hierarchy on the samples that you want to use for the training, right? Which could just be one sample, or it could be multiple in the same experiment if you'd like. Okay, so in this case, I've only drawn them onto one sample, and notice how the other two don't even have a gating hierarchy yet, because what we'll see in the demo is that once these transformations are calculated, it's going to apply those gates anyway. Now, if you have a workspace where the gates are already drawn, it'll just overwrite them. So they don't have to be empty, right? They don't have to be unanalyzed. But the idea here is you would really only need to dedicate a lot of effort to one sample because it's going to group apply those gates anyway. Okay. So up to you. But if you have already an established hierarchy on the other samples, it's totally fine. All right, so with Elasticate, right, I'm going to go ahead and pick my sample. Again, go to the plugins section, which is here in my populations band. Okay, go to, there it is, Elasticate. There it is. All right, here's the dialog window. Okay, so it's going to automatically pick the training sample that I selected. Okay, so I launched the plugin off this population, so it assumes I want that to be the training one. Now it's asking, well, what are the target samples, right? Which samples are going to inherit the gates after we calculate the transformation and apply them? Okay, so you can navigate, if you do have different groups, you can navigate between your groups using these drop downs here, and that'll list all the samples that are in that group in the workspace, and then you can just check or uncheck what you want to have gates applied to. Now, there was a question earlier about, using different training samples. You certainly can. So in this case, I only have one training sample. If I had gates applied on others, I could pick multiple training samples if I wanted to, if I thought it would improve the calculation. So, that's one option you have here over EasyDaffy. You can have multiple training files if you'd like. And then some of you may be thinking, well, I have the training sample and it's also listed on the target sample list. It'll just ignore it. So you don't have to worry about removing it. If it shows up twice, it's just going to ignore that. Okay. All right, so now here it's saying select the gates that you want to apply and adjust, right? So maybe you wouldn't want to do your whole hierarchy, right? But if you do, right, you'll just have to make sure that those all get selected. And if you want to, remind yourself, how those populations were defined, you can select any gate in the hierarchy and it'll kind of remind you, how you did that. All right, now here's some of the tunables, okay? So first one is density mode, right? Default is 1. And if you're ever unsure about what these tunables are, you can, of course, rewatch this video, but also in the documentation, which is a PDF file that comes inside the zip folder with this plugin, right? We break down what each of these settings are, okay? So you can always read it and make decisions about, oh, do I want to turn that up? Do I want to turn that down? So you can always, read this document and it'll help you along with that. Right, so this is the density mode option. Okay, so it's basically, since we're turning these images into pictures, right, we're kind of focusing on the center of populations, right? So if you find that your populations are very major, right, and these major populations are kind of determining where the gate placement should be, right, maybe you'd benefit from turning that number up. I believe it can be from 0 to 3. But if you want to pay more attention to sparse areas, like more outliers, then you would turn that number down. Okay, so you can start with 1 and you can always run different iterations of the tool and see what works for you. But if you feel like your populations are very major and you want to kind of ignore the more sparse ones, and you can maybe just turn that up a bit. Down here, preserve gate type of rectangles and quads. Okay, so here, if you have rectangle gates or quad gates in your hierarchy, having this checked will preserve them. If you uncheck it, then basically the algorithm can attempt to perform more accurately because it's not restricted by these shapes, right? So a quad gate divides the plot into 4 sections with just like a straight line across and the top. And so you're restricted with a gate type like that because you can pretty much only, nudge it so much, and you can't really change the shape of it. So if you turn this off, you give the algorithm a chance to kind of override a quad gate and turn it into something like a polygon, right? And really make it a bit more of a free form shape. Okay. I don't think I have any of these. Well, I do have a rectangle gate type, but so if you are concerned with wanting to keep a rectangle squarish gate, right, you can check that box and it'll do that. If you're not concerned with that, you just want to capture the population, you can uncheck it and kind of let the tool do its thing. Statistic nodes, if you had statistical nodes under each of your gates, right? So if you were here and you'd added a statistic in the workspace, it'll copy that over. Okay, so it could be helpful if you do have a lot of statistics on that sample. And then let's go to the advanced option. So this, I want to skip to the part where there was a question earlier and I want to make sure I come back to it. Where is it? Right here. This toggle right here where it says ignore non-matching populations, right? So somebody asked, well, should I do different trainings, right? What if I have an unstimulated sample? And so I wouldn't expect a lot of, let's say Th1 cells, right? They're not going to be producing a lot of cytokines because they're not stimulated. But I have a stimulated sample, and now I have Th1 population there. So I have two very different looking plots. Like, do I need to do two different trainings? You could, if you wanted to, or you could hit this, this toggle, and it basically ignores non-matching populations. So this could come in handy. Excuse me, if you're in a situation like that, or let's say you have FMOs where, you're intentionally going to be missing populations in your panel because it's missing antibodies for a particular marker, right? So if we turn that toggle on, we're basically saying, if you can't find a population in that same spot, don't try to move the gate to account for that, right? Because I'm expecting it not to be there anyway. Okay, so you could do something like this, or if you wanted to, I mean, the tool is simple enough to run, you could run it off different populations if you'd like to. All right, and then some of the other tools are, a little more straightforward. Here, basically saying, do not allow vertices on the edge of the plot to move inwards. Okay, so if I have a gate that I've intentionally drawn off scale because I want to capture all that data, don't allow it to move back. Like, I want it to stay in that area. Here, optimize image registration for one-dimensional data. So recommended, especially if you have histogram plots, because that's going to be a one-dimensional gate. So we want to run some extra optimization to make sure we capture gates, accurately for histogram plots. Okay. All right, so those are probably the main toggles that I want to point out. So again, a lot to play with. The ultimate goal is to model your data as best as possible. So you may not always get it right the first time, but you can always come back and say, you know what, maybe I should change the density mode. Maybe I should just kind of give up on the fact that my gates can't be rectangles because my data is just shifting too much. Maybe I need to ignore non-matching populations because I've got just too much biology changing between samples, right? So, make decisions that are going to be appropriate for your data and just see what's going to work best. Okay. All right, so having seen all that, let's go ahead and run the tool. So I'll go ahead and say start. All right, so once it's done, see, just take a moment here. Basically what it'll do is it'll say that it finished the calculation, and when I close the window, it's going to apply that calculation to the new samples. Okay, so I'll go ahead and close this window, right? And notice how those two other samples now have a gating hierarchy, right? And you can't even tell that they are, Elasticate calculated because they basically look just like the manually defined ones, right? The only way I know I even ran Elasticate is because I still have the operation node there. Okay, so you can see if I launch that sample, right, and then we can start to, do some inspection on it and see how it did, right, and it did a pretty good job. So I'm gonna go ahead and open up some of these gates so we can take a look at them. Right, so here's all of the, the Elasticate defined gates, and if I go to the next sample, right, we can see how it did there. Okay, maybe it's not doing super great for singlets, like maybe I could fine tune that a bit. And again, maybe I'm a little unhappy with what it's doing with the CTLs, but I could maybe just give up on the fact that I can't have that be a rectangle gate. Okay, but no matter what the placement looks like, at the end, you still get what appears to be a user defined gate, right? I can still come in and make adjustments. I can move the gate however I'd like to. So all the resultant gates are still adjustable. Okay. All right, let's look at the Q&A. What does the number of gray levels control? Oh, that's a good question, Justine. I'm not sure. Let me go back and open that up and see if it's got a tooltip on it. So if I launch that tool, number of gray levels in the images. Let's see. I was hoping it would have a little tooltip here. But yeah, that's a good question. I might have to get back to you on that. I suspect, considering it's next to the density mode, that it has something to do with that toggle, with registering, you know, which parts of the plot to consider. But I can't say I know 100% with certainty what it's doing. Let's see, since I have it open, let's just see if it says it in the documentation. I'm not seeing anything about gray levels. Yeah, that's a good question, Justine. I'll have to get back to you on that one. If it's keeping you up at night, go ahead and send me an email, and that way I'm sure to get back to you, because I'm definitely curious about it as well. But I do suspect it has something to do with the density mode. Maybe it's just a fine tune of that. All right, there's another question. Does the Elasticate allow the creation of template files? Yes, that's also a good question. So yeah, I believe, I haven't tried to do it yet, but I believe, as with any plugin, if you had the operation node as part of the group, it should templatize. That being said, you know, it's pretty fast tool to run already. So, and I'd be reluctant to always, yeah, now that I think about it, you have to pick the training sample to train it on. So I'm not really sure if you would want to templatize it right across workspaces, right, if you don't have that same template sample in there. So yeah, also a good question. I imagine you could make that node group owned and then make that into a template. But I'd probably be more inclined to, yeah, just kind of run it anew in the workspace, now having a new training sample to run it on. I think also, like, as we go with the flow cytometry experiments, right, like the training sample that I pick on one workspace, when I go to a new workspace, like that training sample may not model the shifts that were captured at that particular time in acquisition. So maybe I'd want to pick a training sample in that workspace because it was acquired more recently and it might match the rest of the samples better. But maybe that's just a matter of personal taste. But yeah, I imagine you could turn it into a template if you drag it to the group. But in terms of using it over and over, I myself would probably just pick a new template to run it on in new workspaces. But yeah, it's a good question. All right, let's see. All right, are there any other questions? All right, well, I'm hearing none. So what I'll go ahead and do is just put my email back up here. I know there are 1 or 2 questions that I'd probably have to get back to people on. So if you want to send me an email, I'm happy to follow up with you offline. I know there were questions about publication resources for Elasticate, as well as that kind of density gray area toggle in the plugin. So if you send me an email, I can try to look into those questions for you and get back to you. Otherwise, it was great chatting with all of you today. I hope it was informative. I'll stay online for just another minute if anybody has any last minute questions to add on the chat. Otherwise, I'll go ahead and wish the rest of you a nice rest of your day. And thanks for joining us in today's webinar.